Hello and welcome to today's lesson on testing the effects of antibiotics and disinfectants, where we will conduct a practical experiment to learn more about culturing microorganisms. We're about to head into our lab to explore a fascinating experiment that directly ties into the concepts we've been covering. But first, over to Sam, our lab technician, to outline what safety equipment we need.
Hi everyone, I'm Sam. Before we get started, make sure to put on your lab coat, safety goggles and gloves. Remember, safety is our top priority in any lab environment.
Thanks, Sam. While we're getting ready, let's quickly recap what we've learned so far. So far in our studies, we've explored the fascinating world of cell biology, focusing particularly on culturing microorganisms. We've learned that bacteria can be grown in nutrient-rich environments, forming colonies we can observe and study. Through aseptic techniques, we've discussed methods like streaking plates and using agar to give us clear, uncontaminated cultures. Now, we're moving forward to an experiment that extends this knowledge. Today, we'll be testing the effects of antibiotics and disinfectants on these cultured bacteria. This practical will allow us to observe firsthand how these agents can inhibit bacterial growth and gain a better understanding of how they work to keep us healthy and safe. We're almost ready to dive into the details of our practical. But first, let's explore why this experiment is both exciting and valuable. Understanding how antibiotics and disinfectants affect bacteria is incredibly relevant to our daily lives. Antibiotics are used to treat bacterial infections, which are common illnesses, whilst disinfectants are used to clean surfaces and prevent the spread of harmful microorganisms. By learning how to test these substances on cultured bacteria, you gain insight into how medical treatments and cleaning products are tested during development. The knowledge of how antibiotics and disinfectants work is crucial for public health. Misuse or overuse of antibiotics can lead to antibiotic resistance, making infections harder to treat. By grasping the principles of how these drugs interact with bacteria, you're better equipped to understand the importance of using antibiotics responsibly. This practical not only educates you about microbiological techniques, but also raises awareness about global health issues like antibiotic resistance. Finally, this experiment helps you develop critical scientific skills. You will learn how to culture bacteria, apply antibiotics and disinfectants, and measure their effects accurately. These techniques are fundamental in microbiology and biomedical research. Mastering these skills now lays the groundwork for higher-level scientific studies and research. In summary, this practical is not just an academic exercise. It's an exploration of real-world applications, public health implications, and scientific methodology. Now, let's dive in and see how antibiotics and disinfectants influence the growth of bacteria. On to the practical. Let's start by gathering our materials. Sam, what will we need?
So for this practical, we need a Petri dish, a bacteria sample, paper discs, beakers containing antibiotic or disinfectant solutions, a ruler, an inoculation loop, forceps, an autoclave or a Bunsen burner, and an incubator.
Make sure your lab area is tidy and all your materials are within easy reach. We begin with our preparation stage. For this practical, it's crucial to keep our setup organized and maintain sterile conditions to ensure accurate results. We must therefore sterilize all our equipment before we begin, including Petri dishes, forceps, and inoculating loops to prevent contamination. We do this by using an autoclave or by passing each piece of equipment through an open flame using a Bunsen burner. We also need to prepare our agar plates. Using a permanent marker, divide the bottom of the agar plate into 4 sections as shown in the diagram, marking a dot to indicate where the discs will go and labeling each section. Once our preparation is complete, we can move on to step 2, inoculation. Using a sterile inoculating loop, transfer a sample of bacteria onto the surface of the solid agar in each Petri dish. It is important that when we use the inoculating loop on the Petri dish, we spread the bacteria evenly to ensure consistent growth. Next, we need to apply the antibiotics and disinfectants. First, soak small sterile paper discs in different concentrations or types of antibiotics and disinfectants. Ensure each disc is thoroughly saturated in the antibiotic you're using. We also need a control disc. To do this, soak one paper disc in sterilised water. Now, carefully place each soaked paper disc onto the surface of the agar plate. Use sterile forceps to avoid contamination and ensure each disc is pressed gently onto the agar. There are a few important things to remember during this stage. Firstly, ensure to keep a Bunsen burner nearby so you can continually sterilise the forceps used to place the paper discs on the agar plate. Secondly, we must remember to ensure that the lid of the Petri dish is only partially open, even when we are placing the soaked discs on. This prevents any unnecessary contamination. In our next stage, we need to incubate the agar plate. Before we place the agar plate in the incubator, we need to ensure that it has been taped properly. Do not seal all the way around the lid, as this prevents oxygen entering and encourages the growth of dangerous anaerobic pathogens. Once partially sealed with the tape, position the Petri dishes in an incubator set to 25 degrees Celsius. Although bacteria can grow faster at higher temperatures, 25 degrees helps to balance growth speed while ensuring safety. Leave the dishes in the incubator for 48 hours to allow bacteria to grow and for the antibiotics or disinfectants to take effect. Now for our fifth and final stage, after the incubation period, carefully remove the Petri dishes from the incubators. You should be able to see clear zones around each paper disc. This is where the antibiotics on the soaked discs have prevented the growth of bacteria. Using a ruler or caliper, measure the diameter of the clear zones, known as the zones of inhibition, around each disc. These zones indicate areas where bacteria have been killed or prevented from growing by the antibiotics or disinfectants. When you observe the Petri dishes, you'll see clear zones forming around some discs where the antibiotic or disinfectant has prevented the growth of bacteria, highlighting the effectiveness of various antibiotics and disinfectants. Let's now analyse this experiment. The central biological reaction in this practical is the interaction between antibiotics or disinfectants and bacterial cells. Antibiotics either kill bacteria or inhibit their growth by targeting essential bacterial functions, such as cell wall synthesis or protein production. Disinfectants, on the other hand, typically act by disrupting cell membranes, denaturing proteins, or disrupting other essential processes within bacterial cells. To analyse the results, follow these steps. Firstly, measure the diameter of the zones of inhibition and record each diameter in your lab book. For each paper disc, measure the diameter of the clear zone in millimeters. Ensure measurements are taken consistently across all samples. Secondly, calculate the area of the zone of inhibition. You can use the formula area equals pi r squared to find the area of the zone of inhibition, where r is the radius, half of the measured diameter. We will see some examples of this in our next lesson on culturing calculations. Next, we need to compare the effectiveness of the antibiotics and the disinfectants. Create a table to organise your data with columns for the type or concentration of antibiotic or disinfectant, the diameter and area of the clear zone, and any observations. Note that if multiple tests were performed, we need to calculate the average diameter and area of the zones of inhibition for each type or concentration of antibacterial agent. Conducting multiple experiments and taking the average like this reduces the influence of any outliers or experimental error. Now, we take our data and plot the average area of the zones of inhibition on a bar graph. Use the different types or concentrations of antibiotics or disinfectants on the x-axis and the average area of the inhibition zone on the y-axis. This visual representation helps in comparing the effectiveness of each agent clearly. Finally, discuss the results, noting which antibiotics or disinfectants were most effective and why. Consider biological mechanisms, such as the mode of action of each antibacterial agent. In our example, antibiotic C has the biggest zone of inhibition, therefore it is the most effective. Antibiotic B has the smallest zone of inhibition, therefore is the least effective because it killed less of the bacteria on the agar plate. In your discussion, you should also identify any unexpected results or anomalies. Consider reasons for these, such as potential contamination, uneven bacterial distribution, or variability in the concentration of the soaked discs. This structured approach will help you precisely understand how different antibiotics and disinfectants affect bacterial growth, and make informed conclusions about their effectiveness. That's the end of our lesson today. Let's recap. In today's lesson, we conducted a practical experiment to investigate how the effects of antibiotics and disinfectants on cultured bacteria are tested. The key steps of this practical were preparing agar plates, inoculating bacteria, applying antibiotics and disinfectants, incubating agar plates, and observing and measuring our results. One of the key takeaways of this practical is the importance of using sterile techniques to avoid contamination and obtain accurate results. More information on this is in our lesson on aseptic techniques. We also learnt the importance of precise measurements to record zones of inhibition, as well as understanding the use of controls to validate experimental findings. This practical not only reinforced our knowledge of how antibiotics and disinfectants function, but also highlighted the critical techniques involved in microbiological experimentation.
Watch the rest of the GCSE biology course at Sophos Education.